When we look at a leaf's surface, we see its green color and smooth texture. But if we try to look closer, to find the actual living structures keeping it alive, our eyes hit a physical limit. The tissues that build plants and animals are too small for the naked eye to resolve. For a long time, education relied on the passive memorization of two-dimensional diagrams in a textbook. However, biology is an active science that requires physical evidence. To understand living organisms, we must find a way to reveal their hidden architecture and mathematically determine their dimensions ourselves. The light microscope is the primary tool for this investigation. Operating it effectively requires a specific sequence of masterable physical skills. The investigation begins with the shift from viewing diagrams to physically preparing a specimen for measurement. The laboratory environment is designed for this process, providing a space where raw biological material is systematically converted into readable data. The first step is extraction. Using tweezers, we peel a layer of tissue that is thin enough for light to pass through it completely. We lay that tissue flat on a glass slide. Because many internal structures are transparent, we add a drop of water or a chemical stain to provide the necessary contrast. Next, we lower a cover slip at a 45-degree angle. This specific movement sweeps the liquid across the slide, pushing out air bubbles that would otherwise obscure our view. Once prepared, the slide is placed on the microscope stage and aligned directly over the light source. Following a strict protocol, we always begin on low power. This wide field of vision makes it easier to locate the specimen and establish its position before moving closer. After centering the target, we switch to medium power. This magnification reveals the individual cellular boundaries that were previously hidden. Through correct slide preparation and progressive lens adjustments, an invisible sample becomes a structured cellular landscape. Looking through the lens creates a challenge. The cells now appear quite large, even though we know the physical sample is microscopic. We need a way to determine their real-world size. To solve this, we use the field of view. This is the actual physical diameter of the circle visible through the eyepiece. We measure the field of view's diameter with a millimeter ruler. If 10 cells fit across 2 millimeters, we estimate 1 cell size. The magnification formula is then the size of your drawing divided by the actual object size. If your drawing of a cell is 20 millimeters long, but the actual cell is only 0.2 millimeters, the math proves that your drawing is a 100 times magnification of the original specimen. By adding physical measurement and calculation to our observations, we convert a visual image into verifiable scientific data. We have now completed the sequence, moving from a raw piece of tissue to a specific, calculated mathematical value. A student who relies on being told the answers remains a passive observer. However, a student who prepares the slide and performs the math has the tools to verify their own findings. These skills, from slide preparation to mathematical calculation, allow a researcher to produce repeatable and verifiable results from a raw specimen. Combining slide preparation with the magnification formula allows the learner to move beyond simply seeing a cell and begin quantifying the actual physical dimensions of life.